So this is the last panel of the day. Last year, we had a very different structure. We really had companies presenting with one key opinion leader. And this year, we had really key opinion leaders on panels. And so I thought very carefully and long about what panel I wanted to moderate. And I'll tell you that in many ways, I concluded that this panel, the regulatory landscape and the role of manufacturing, was the one I wanted to tackle. I also believe that in many ways, this is one of the most complex panels you could possibly have. And it's in part because manufacturing and the regulatory pathway is so critical in the cell therapy space. It's new, it's novel, the half-life of the drugs are different, and so the idea was to really put together a panel that could help us understand what it's going to take to get the first products approved and make them commercially successful. In other words, what we really want to understand is how is cell therapy different than biologics? And maybe it's more similar to biologics and monoclonal antibodies than it is different. So I went through the FDA website and I started looking at what the FDA is saying in advance of this, and it became clear to me that the FDA hasn't really updated their website in a long time, because some of Joyce, I know, is shocked at that statement, having spent 12 years at the FDA, and particularly in this area. But it's really interesting to me because the FDA website talks a lot about extreme levels of caution and a very different mindset. So one of the questions that I have as an analyst, because I'm not really inside on the FDA, is how up to speed is the FDA with some of these differences? And we'll talk a little bit about this. What are the regulations on stem cells? And of course, this isn't just a US question. We're so excited to have Heki-san come from Japan because we've all been hearing so much about new regulations in Japan. And I realized in the telephone interview that I did with Heki-san last week that I got it wrong. I was very, very excited by the new regulations in Japan, and I really believe that the new regulations would represent a fast track to the marketplace. But when I started digging for what those regulations were, I couldn't find them. And I looked everywhere. I have a Japanese wife at home, so we did a search in Japanese, and I still couldn't find them. And interestingly enough, in my conversations with Heki-san, we talked a lot about what are they, what are the details. So hopefully we'll get some insights into what that means. So these are the questions that we're after today, and it's understanding when we're dealing with cell therapy, what is the dose? How long do the cells last? How long do you have to give them? We heard Dr. Pecora talking a little bit about the threshold effect. So how do you define a threshold effect when you're dealing with cells? How is it manufactured? Is the source itself safe? And we talk about mesenchymal cells. We've talked about autologous cells. We've talked about allogeneic cells. But we're going to start to hear more about multipotent and pluripotent cells. And what does that mean in terms of therapeutic value indications? How do you expand these to the commercial scale? In one of the conversations I had with David Smith, we were talking a little bit about cell banks, and David was correcting me that that might not be the right word to use when we talk about cell therapy. So we'll hear a little bit about that. How will these products be delivered? You know, who's thought through exactly the process and how it's delivered? What do the regulators want to see? And of course, what are the implications here globally, and particularly in the US, Europe, and Japan? So our panel is very exciting. I'm going to call up David Smith of Lonza, who's going to introduce us a little bit to the nuances of manufacturing. Greg Rosati of Celgene will follow and kind of segue a little bit. Dr. Joyce Frey will talk a little bit about the FDA and her consulting experience as a ex-FDA reviewer. She spent 12 years there. And Tatsuo Heki-san will talk a little bit about what the cell therapy and regulations are in Japan. Okay, I look forward to an exciting panel.
For you who don't know who Lonza is, we're a large contract manufacturing organization based in Basel, Switzerland. And on the cell therapy side, our expertise is really around large-scale allogeneic processing. We produce the bulk of the cells that are in the clinical trial today on the allogeneic side. And I asked myself, what could a contract manufacturer tell you about regulatory path? Well, what I'd like to assure you is that while cell therapy's path forward may not be totally clear. It may be still, this road still needs to be built. It's not a road that hasn't been built before and built successfully, and companies you probably invested in. Go back to 1993. Lonza bought a little company called Celtec out of the UK. At that point, we had very inefficient, they were the emerging contract manufacturer for proteins. This is a mammalian company. They had very poor cell banks at those time. They had upstream processing where they used large amounts of serum to get very low yield. They had downstream processing that was crude, and they lost a large percentage of their drug product in the actual downstream process itself. And their output, they were excited by the concept of micrograms or milligrams per liter of media. They had many regulatory issues that came forward. You know, in the late 90s, they figured out that you couldn't scale up from a 1,000-liter reactor to a 20,000-liter reactor because the proteins didn't act the same when you came off of them. They found PAS. With the help of a lot of researchers who were out there, a lot of the tool companies, they were able to work together and come up with a solution moving forward. In 2007, Lonza acquired a company from Cambrix, which was the cell therapy organization. They believed that cell therapy was the disruptive technology for their protein industry down the road. And they felt they needed to be in early and they needed to be aggressive. Where were we at in 2007? We were excited by the concept that we were getting our first closed processing cell factory work. We were using downstream processing that was basically stealing technology from the blood industry. And we were incredibly excited because for the first time we were doing more than 2 billion cells per lot. And we had the same media problem that they had back in the Celtec days. We were using boatloads of serum, fetal bovine serum, for every single dose we were producing. Again, not well characterized, not well moving forward. So how did we do it? Lonza, first thing we did is, Lonza, this is an older slide because it was important. We did our first strategy for Lonza in 2009, where we actually looked at how we were going to do upstream culturing. Again, it was called cell culture, and upstream and downstream, because they thought like protein people. But what they were able to do is they brought in process engineers who landscaped out a roadmap that looked at what will these companies need to produce to be successful in 2020. What is the scale of the processes? What are the scale that we need to get to? What are the cost of goods we need to get to make them successful? And I have to tell you, we wrote this in 2009. We're actually on track with this. And we also looked at the downstream side. Again, we can't lose large amounts of cells in the downstream process. So we've been working with a number of companies, partnerships, acquisition, teaming up, and actually stealing technologies from other parts of industries to actually find new technologies to get the highest yield possible, again, driving the cost of goods. What's the real regulatory issue to this? Yeah, we made this beautiful roadmap. It's great. However, the technology requires shifts. Early phase, where most people come in at research or early phase, we do the first step. It's very straightforward. We close up their process. We make it a closed processing system. We scale it up in planar technology. We can take them from that first S-curve up to the second curve where we can get much larger doses, much number lot size, because lot size drives cost of goods. The issue is that to make the next leap, the current technology, the planar technologies that are out there today will allow you to get to a certain scale, early commercial, small indication commercial. But when we talk large indication commercial, we need to make a theoretical jump into the third curve where we're talking about true commercial scale, where we're talking about thousands to tens of thousands of doses per lot. How does that get achieved? We achieve this by really, first thing is focusing on the characterization. We drive this message to our customers on a regular basis. We're developing technologies to look at it, and we're partnering with people to come up with that. These technologies are still in the developmental phase, and it's interesting. The one thing we ask people to do is tell us when your process fails. In general, people walk in and say, I have a process, it's never failed. Well, then you haven't characterized yourselves. So we force people to think about, how do we fail? Second, focus all your dollars on the things you can't change later. One of the things that Greg will talk about is cell banks. These are not forever cell banks, but quite often people don't push their cell banks out to the most passages possible because they're going to need them later on. They don't choose media with the thought of growth factors that are going to be available, and what are the scarcity issues, and what will drive down your costs long term. Again, issues that will change things now. And the other one, which is kind of interesting, and we see it all the time, they don't choose a package that is commercially viable. They choose a package that works in research, is easy to do in the research setting. However, when I have to ship 20,000 of them, 200,000 of them, is it the appropriate dose? And you have to then make that shift at that point and back up and start your shelf life studies again. The last thing, and I think Joyce will kind of talk about this more, is really process validations. Today, there's not good process validations. It's going to be early, but it's been done. The same process validations that we're starting to work with our phase 3 customers with are the same ones we took over from the biologic industry. It's the same thing. So if I could conclude by just saying, yes, the roadmap to commercial, regulatory and commercial success for cell therapy isn't totally defined yet. It's still working through. However, it's a road that's been produced and has been successful in the past. Thank you.
Terrific. Dr. McLeod? Thanks, Dave. So I want to start by saying I completely agree with Dave that this road we're traveling down right now in manufacturing cell therapies is very similar to the road we've traveled successfully down for biologics. There are some differences, however, and I want to allude back to a point Dr. Pecora made a little while ago about if you have a therapy that works, the manufacturing, the supply chain, everything else will follow. However, I think there's great risk in that if you don't do the manufacturing well, if you don't understand your process and your product well, you will fail in clinical trials. And there are a couple of challenges specific to cell therapies that were not common to biologics. And I'm going to hit on one of them today, and I can talk about others as well if we get to it. And I think what's key to these things, again, is having a strong understanding of your product and knowing how you can address certain changes you might have to make. So one of the examples I just want to hit on briefly today is this idea that Dave set me up for as far as donor comparability. I'm going to speak about this in the context of allogeneic cell therapies. And when you think about changing banks or changing donor stock, you think about other changes. And there are certain changes that, as Dave said, you want to make. You want to do things early that you maybe can change later, but there are things you have to change later, such as scale or maybe the other characteristics of your product to make it more potent. So this slide just depicts the fact that as you go forward, you want to evolve your process less, but you have better tools and a better process understanding to make changes. And some changes will be inevitable. And the key to all of this is understanding your product and understanding potency. So within that context, let's think about cell banks. For biologics, you have the advantage of having great proliferative capacity of your cells. So for a monoclonal antibody, your CHO cells, you can make as many as you want. You have great proliferative capacity. So you can make very large banks, and you typically do this to last the lifetime of your product. And you have a two-tiered banking system. And if you change the master cell bank, you're probably doing it for a really good reason. And that actually might mean you have a different product, maybe a different glycosylation pattern or something different about the potency of your product. And that could be a big change. And we look like we lost the slide. Oh, there it is. OK. So with cell therapies, you want to make big banks as well. But the proliferative capacity of your cells may not be there to have a bank large enough from one donor to treat all the patients you're ever going to treat. So you do this in a way that's two-tiered. You do this in a way that you have consistency among the different vials of a bank. But even if you have doses or banks on the order of making doses, 100,000 doses per donor, you're still going to have to change at some point depending on the market and how much you dose your patients. And when we spoke to the EU regulators at the Committee for Advanced Therapeutics, they actually said, don't call them banks. That has a connotation that you're never going to change it. So we call them stocks, and we have a good plan as to how we're going to have comparability of these stocks. And that plan, they did say you can call it a bank, and you can consider it a different product if you want to and file a new MAA every time, but I don't think anybody's interested in doing that. So you need to have a plan on how you're going to do this. So there's some obvious ones. Obviously, when you change donor stocks, your release specs need to be met. You might have other characterization tools that you'll want to use to show your product is still consistent with the product you made from the previous donor stock. Stability is obviously very important. But when you first start doing this and you don't have a great handle on efficacy, you probably have some in vivo measures, some animal studies you're going to do to give you confidence that the cells have the right efficacy, the right functionality. So you're going to certainly do those. And ultimately, you need to tie it all together with clinical data. And as you start out clinical trials, obviously you want to make sure safety is maintained. As you go forward, then efficacy becomes extremely important as well, both from a regulatory as well as a business perspective. So then the question is, I just want to frame this out as how you go about doing this. When would you bridge from donor to donor? So if you just think about a very simple clinical pathway, you can do this early, and I know there's examples of companies that have used multiple donor stocks early on, but your patient numbers are going to be so small, you'll have no idea if there's a difference from one donor stock to the next. So then you can do things in a more controlled fashion early on. Maybe you set up a separate phase 2 trial, or within your phase 2 trial, you build in more patients to examine donor-to-donor differences in a controlled manner. But this would be extremely expensive to do so early on. It's a big pre-investment when you don't even know if your product's going to work. So do you wait? You can wait till phase 3. Obviously, there's risk there. You could do it in a controlled fashion in phase 3, where you have a parallel trial just to look at that. But the patient numbers you're talking about there will lead to large expenses. If you think about phase 3, however, if you understand your cells very well and you understand potency very well, then you could potentially design a trial that builds this variability in without looking at it as its own variable. So that's an option. Or if your bank is big enough, your donor stock is big enough, and it's enough to get you through launch, maybe you defer this question until later on when you know you have a product, and post-licensure you do a trial to bridge you to other donors. Regardless of what you do, you're going to need a good comparability plan. This is, again, one example of a change that in many cases for an allogeneic you have to make. Unless you understand your product and understand your process and have good assays and good tools, there's a really, really big risk that you're going to fail in your clinical trials because of your quality and because of your manufacturing. I'll just close by saying, when we think about our regulatory approach in terms of manufacturing, there certainly is a regulatory landscape. A lot of it was adapted from the world of biologics or other types of therapies. Some of it doesn't always fit because every cell therapy is different. MSCs are different than CAR-Ts, or different than NKs, or different than other types of cells. But we understand what the landscape is. We use data to drive our decisions. We've been very fortunate. The FDA, CBER at the FDA, and specifically the Office of Cell and Gene Therapy is very data-driven. The EU regulators we've talked to have been very data-driven. So if you use data, work within those regulatory constraints, we feel like we can define the regulatory landscape as we go forward and make good decisions based on that. And with that, I'll turn it over to Joyce.
I can figure this out. Okay, I'm going to go through some of the newer things that are out at FDA and kind of give you a little few hints as to why it works in some situations and why it's not working in another. And what I did on this side is try to compare and contrast FDA and the EU. And one of the new things in the US, which appears to be going to happen in the EU, though it's not real clear, is a breakthrough therapy designation. Though I'm not sure how successful it's really been. Right now, FDA's received 159 requests. There's been 41 granted, 87 denied. And out of the ones that have been granted breakthrough, there's actually been 4 approvals. They've all been in the Center for Drugs. The all the approvals have been. There have been only 15 breakthrough therapy designation requests to the Center for Biologics, and only one's been approved. I do know from my internal connections, there have been at least 10 or more breakthrough therapy designation requests sent to the Center for the sent to the Cells, Tissues, and Gene Therapy group. None of them have been approved. Oh, I went the wrong way. And part of that is really the definition of what breakthrough therapy is. When Congress put this in, it was great for investors, for companies and working with investors and getting investment. But I think people have misunderstood what it was really all about and the criteria to meet it. It's a very high hurdle. You really have to have preliminary clinical evidence. And that really dictates in totality whether you're going to get breakthrough therapy designation or not. This just lists some of the features, but some of the problems that FDA is seeing is when you think about breakthrough therapy, the entire designation is strictly on your clinical package. They don't look at your CMC at all. And what's happening is FDA is finding that the manufacturing is not ready at the same pace as the clinical. And at least in the US, if your manufacturing is not up to snuff as your clinical, you're not going to get approved. Now, the whole purpose of breakthrough therapy was really, it's to improve the communications between you and FDA. Personally, coming from the Office of Cells, Tissues, and Gene Therapy, I don't think breakthrough therapy designation is going to get you any advantage with that office. They talk to people all the time. And they always have high-level management at any meeting you attend. So I'm not sure how beneficial breakthrough therapy designation is for cell therapies, other than, like I say, for some reason, the investment community thinks it's a big deal. This is a slide that lists the approved products, both in FDA and EMA. I put it from 2007 because that's when the advanced therapy in EU came on board. There's one more in the US, and that's Carticel that was approved. And I've given you the year as well as what mechanism they were approved under. And as you can see with Glybera, Unicure, it was approved under a very unusual approval pathway, and this was the one case where the CAT recommended approval and the CHMP, who has the final say, was not willing to approve it. So it was kind of a fiasco for the EU. The approval pathways, in the US, you have full approval or you have accelerated approval. In the EU, they have a few additional approval pathways, and so does Canada. And so they have full approval, they have conditional approval, they have approval under exceptional circumstances, and they have accelerated approval. This is a slide that compares the different approvals, standard and priority. You both, in either one of those pathways, you get full approval. It's the accelerated approval that's the different one, and what differentiates that is really your endpoint. It needs to be based on a surrogate endpoint. If you have a clinically meaningful endpoint, you're going to be a standard or a priority approval. This just goes through some of the additional features of the different pathways. And then this slide lists the EU pathways. So the standard approval in EU is 210 days. There's 2 clock stops. Then you have conditional, which is based on incomplete clinical data. There have been a couple of approvals in Europe based on that. The approval under exceptional circumstances is the one Glybera was approved on. Canada's approval pathway, very similar to the exceptional circumstances, is the one where the Osiris Prochymal was approved in Canada under. But the EU also has accelerated approval, though they have very clearly said that this needs to be justified and this should only be rarely used. Well, no, it's exceptional circumstances. It's very rarely used. And then they also have compassionate programs. So when you think about the approval pathways, you really need to think about where you're trying to get approval, what regulatory agency you're going through, because every regulatory agencies have different pathways, and there's different conditions for all those different pathways. What works in Europe, what may work in Japan, we'll see how that pans out, is not going to work in the US. And so you really have to think about, as part of your strategy, what is the approval pathway and what country can I leverage? And that's it.
First of all, thank you very much for giving me this opportunity to speak. I work for Fujifilm, but I represent FIRM, which is the Forum for Innovative Regenerative Medicine. I just would like to show one slide of introducing what FIRM is. It's close to ARM. It's 76 companies. We represent the industrial sector when there is some kind of a medical discussion panel on the government. And right now, as you can see, the companies in FIRM are not only the companies to cell therapy, but there are machinery companies, biotechnology companies. Like for machinery companies, there are companies such as Hitachi, or those kind of large electronic companies. So a little bit different from ARM is that FIRM, most of the companies are large Japanese companies who is seeking for new business in this regenerative medicine field. And also Fujifilm is one of the companies that is in the similar position. Now I would like to speak about the change in regenerative medicine in Japan. First of all, there are three laws or acts that we should consider. The first one came out last May, which is the Regenerative Medicine Promotion Law. It's a kind of a law that asks the ministry to promote the regenerative medicine. And it allows you a lot of money to use it. So that's the first law. What the audience here would be interested is about the two new laws for the regenerative medicine. And these two laws are independent. One is the Act on Safety of Regenerative Medicine, and the other is the Act on Pharmaceuticals and Medical Devices. And usually, people, my impression in the United States is that most of the people look at the change in the Act of Pharmaceuticals and Medical Devices. But I would like to speak a little bit more about the first law, which is also an innovative law, I think. The background why these laws came out is that there are guidelines for regenerative medicine, but it was not based on law. So it was a desire or it was a necessity to make a law to cover the regenerative medicine. And next is that there is a tendency that there is a need for collaboration between the medical institutions and the industry for the early stage of development for the new development. These two are the backgrounds that we have a new legislations for promotion of safe regenerative medicine. This is the Act on Safety of Regenerative Medicine, the first law. The second is about the change in pharmaceuticals affairs laws. First of all, Fujifilm is in the medical device industry. It was very strange that there is a division called Pharmaceuticals Affairs. This is a history that medical devices were regulated under Pharmaceuticals Affairs laws. That was Japan. But now the government find out that this is so strange, so they changed Pharmaceutical Affairs laws. And on that occasion, they introduced a chapter dedicated to regenerative medicine. Going back to the first law, the Act on Safety of Regenerative Medicine. This is a structure where, first of all, it's a relationship between the hospital, and the new concept is that the hospital can commission cell processing to an outside cell processing center. And this was done by, first, the hospitals are obligated that they submit plans to the regulators. And then they commissioned the cell processing center for cell culturing. But the cell culturing process center has the obligations to notify that they have the right, or they comply to the requirements that is made by the regulators. And these kind of structures made it possible that this is not a approved medical device or approved pharmaceuticals, but it realizes to actually do a regenerative medicine therapy based on the structure. This is one scheme that the Japanese government has introduced. And of course, based on the risks, the approval of doing the therapy is, if it's a high risk, it goes to a very higher ranked approval. But if it's a low risk, it's more easier. That's the scheme. And this is the first brief explanation of the left side, the Act on Safety of Regenerative Medicine. Then I'll go to the explanation of the right side. This is the change of the Act of Pharmaceuticals and Medical Device Act. The change is two. Number one is that, as I mentioned before, we have a new chapter for regenerative medicine. It's an independent chapter dedicated to regenerative medicine and cell therapy. The second is that what they call the expedited approval system for regenerative medicine products. This is usually written in documents as saying conditional approval process. The point is that if you wish to acquire approval, usually you need sufficient data for safety and efficacy. But in this case, you only need, they say, assumed efficacy is the terminology that they use. This document has been provided by the Ministry of Health, Labor, and Welfare. So I think they had a very hard time in translating Japanese into English. But what they say is assumed efficacy. And then you can get approval, a conditional approval of a regenerative medicine product. But there's a timeline after that. After marketing for less than 7 years, you have to get a kind of a full approval of the product. That's the heart of this act. So this seems to be very beautiful. And actually, these two laws are a step forward for regenerative medicine industry or the participants who are interested in regenerative medicine. These acts will become valid this November. And what the government, in cooperation with the industry, is doing right now is preparing what we call the guidelines for these acts. The regulators need guidelines to make decisions. And that is the point where whether this makes it very easy for producing regenerative medicine products or not. At least this is one step forward, but we do not know how much that step is large. It could be a small step, but when the regulators become very conservative, it could be a very large step when the regulators understand what regenerative medicine is and make, I would say, more appropriate decision for the industry. So at least we will find more this November when we look at all the guidelines. And of course, after that, when we see the examples of approvals, then we can learn more about how the regulators will make decisions on the regenerative medicine products under this new act. One final slide I would like to show you is that this is my expertise. Actually, I would like to inform you that in ISO TC 273 biotechnology, we now have a task group going on to consider or to work on the standards for regenerative medicine. For example, TG2 is going to make standards, international standards for biobanks and bioresources. TG3, this is going to be led by the United States. This is analytical methods. They're going to seek standards to measure cells performance, cell performance. And TG4 is bioprocessing. Actually, I'm the leader of it. And we intended to make standards to help the bioprocessing for the regenerative medicine. The scope of the 276 is more broad, but the main focus, I believe, would be on regenerative medicine. And Japan wishes to have good cooperation with the United States people in this field. So that's my presentation. Thank you.
Heki-san, thank you. That was very good. Help me understand a little bit. What was the feeling that kind of drove the need for this legislation? I think here in the United States, some people believe that Japan was trying to attract foreign companies in the regenerative space by essentially creating a fast pathway. But what I hear you, is that true? Yeah.
Actually, one of the things that is missing is that when you look at the Japanese industry usually is good at doing tooling, like making devices for measurement or having constant quality control for the raw materials. So the Japanese government, especially the Ministry of Economy, Technology, I'm sorry. The Japanese government, who wished to promote Japanese industry, thought that this is a new area where the Japanese industry can have a strength. So that's a background, one background.
Good, thank you so much. And one other question about Japan and the new regulations. Just to summarize what I think I heard you say was that they've kind of announced that there's going to be new regulations and a new pathway, but that we really don't have the details yet. And I think I heard you say that in November, we're going to get our first glimpse at maybe what the draft guidelines would be. Is that correct?
Actually, to be more specific, by November, they have to have all the guidelines.
Okay, it sounds like you don't have so much confidence that we're going to have all the guidelines.
Yeah,
Do you
they
think we
should
will?
have it, and they are quickly making it. That's the status right now.
Okay, terrific. So let's kind of work our way backwards. So Joyce, let's talk a little bit about the FDA, and help me understand. I know that you deal with lots of clients. You and I had a chance to interact a few years ago on the Amersite product, and so I know you have tremendous expertise in this area. When you're consulting with a company, are they taking into consideration, or how does the FDA take into consideration things like dosing, or we were talking a little bit about cell stocks. How does the FDA look at the manufacturing section of a cell therapy versus how they might look at a monoclonal antibody? Do they perceive it as different or no?
Well, what they look at it differently is, there's certain regulations that, for approval you have to follow. They're the law. And the FDA can't deviate from those regulations. They've been sued over it. They're not happy about that. So it's how you approach those regulations. So when they look at things like cell banking, the multiple donor issue, if you know you're going to have to use multiple donors, they would like you to do that during your clinical trial. It's not a showstopper. They're not quite as the way EU is. It says, well, it's a new marketing application. They recognize that for cell therapies, there's going to be differences. I think you're going to find that there's a lot of flexibility built into this particular office. And they look at things, they understand that there's regulations they have to follow, such as process validation. You have to do process validation, which, quite frankly, when that guidance document came out, the cell therapy industry said, oh, it doesn't apply to us. But in paragraph 2, it clearly said it applies to cell therapy. So don't think just because you're a cell therapy, certain regulations don't apply to you. They all apply to you. It's just how you approach them.
And Greg, can you share with us a little bit about your experience, in terms of the decision making on this issue? Is this something that, in these early clinical stages that Celgene is in now, phase 1, phase 2, how do you deal with the stock issue?
So I think we made a decision early on to not make that a variable in phase 1 or early phase 2 because there's enough things you're wrestling with at that point that if you add that variable and you don't understand enough about what makes your product potent and what might make one donor stock different than the next, it's foolish to do that early. So we now are getting a pretty good handle on what makes our cells work, and we're picking the right time to bridge donor stocks based on that understanding and confidence that we're not going to take a bad risk and lose the initial efficacy that we're seeing.
And David, as a supplier in the allogeneic world, I know that we were talking a little bit about Lonza and some of the companies that Lonza works with. How when you look at the allogeneic world where this becomes a bit, obviously it's a critical issue, how are you dealing with this with companies like Johnson and Athersys and Mesoblast, and how do they approach the idea of sources and cell banks or stock?
So everyone at this point, as they approach phase 2, as Greg said, phase 2, we actually recommend, we have built a fairly robust tissue sourcing group for them where the FDA has actually reviewed some of the documents around that, around donor histories and informed consents, and very tied up. And we will recommend for them very early on to actually produce 3 to 5 cell banks right out of the box. It's not that expensive to produce them. It's expensive to test them. So we will have them produce multiple ones, maybe only choose from the research side 1 or 2 to take forward. And then you have, by phase 3, you already have defined your processes, and you have enough to choose from so you can see the variability between the 3 or 5.
Can we explore a little bit about whether there's any bias towards autologous or allogeneic? It seems that Lonza is very focused on the allogeneic model and not on being a contract manufacturer for the autologous model. Is that true?
I think that we continue to do some autologous work, but I think if you look at Lonza's expertise, where they can bring the most biomanufacturing expertise into it, it's on the allogeneic side. We think in batch production, and we think about how large we can get to lots, what's the lowest cost we can get, and that's our focus. We continue to do autologous. We just think it's a market that is yet to be defined. And companies like GE are going to work very hard to kind of further tie out those marketplaces.
Is it, Dr. Bakura made a statement earlier that the costs between autologous and allogeneic are both very high, but what I thought I saw you presenting on your slides was that eventually you can engineer a pretty low cost of goods associated particularly with an allogeneic model. So do you feel confident that that's true?
We've seen the progression from years ago. I can tell you we were in $10,000, in 2002, 2003, we were at $10,000 a dose. We have dropped the magnitude from that point. We continue to drive processes where we look at those hundreds of dollars per dose. And I think at that point, our view is, as you get these very large doses, then you can attack these indications that are much larger and really become a true broad disruptive technology.
And Joyce, when you're looking at autologous versus allogeneic in terms of the FDA, is there a different set of requirements that they'll perceive associated with that? For example, safety? No. So
the only difference is between autologous and allo. It's really, with autologous, you don't have to do the human pathogen testing. Right. If you go to Europe, you still have to do the human pathogen testing on autologous products.
Which doesn't make any sense, right?
Well, I know, but it's a difference. And in Europe, it depends upon what country you're going into as to what pathogens you have to test for. It's a very difficult landscape in Europe when it comes to cell therapies.
And how about in Japan? Is there a bias towards autologous, or do you think that the Japanese people as a country, as a culture, will be comfortable with the concept of allogeneic cells as a therapeutic?
Right now, most of the people are more interested in autologous right now. But when we think about businesses, autologous might be difficult. So as for a future kind of a plan, we have to shift it. That's the industry side thought.
So it's kind of a cultural bias that autologous is safe, and the idea of allogeneic is culturally, not so desirable unless you show kind of definitive efficacy.
Yeah, I would say cultural. Well, you can say it's a culture, but we're still learning how difficult it is for allogenic.
And talk to me a little bit about, we know that in Japan you have the highest incidence and prevalence of stroke in the world. So we know that Athersys is going to have phase 2 data coming later this year on stroke. Is it possible that, Athersys would be able to access a pathway? I mean, is there an awareness in COSATIO of a need to treat stroke, and that's kind of part of this regulation strategy? I know that's kind of...
Yeah, I'm sorry, I cannot comment on that.
Yeah, no, it's a kind of open question, I know. Very good, thank you so much. So let's just talk a little bit, in terms of the regulatory landscape, and people were touching on endpoints a little bit this morning. And, we listened to the cardiology panel. Are the endpoints discussion any different for cell therapy than it should be for a monoclonal antibody or a complex biologic?
Right now, it has not been. I think FDA is looking at it. I think they realize that, particularly when you regenerate tissue, that maybe the endpoint shouldn't be different. But there again, you have to understand that their regulations say a clinically meaningful endpoint. And so you have to try and connect those dots. And I think sometimes when people go and want to propose different endpoints, they haven't, what I'm finding is they haven't connected those dots. And that's what's happening, I think, even with the breakthrough therapy. Number one, with cell therapies, people are going in with very small trials. They're submitting their requests after they've done a small phase 1 study. The smallest breakthrough designation I have seen to date has been a trial with 43 patients. FDA is not going to give you breakthrough therapy when you go in with a 10-patient phase 1 dose escalation study where you've treated 3 patients, 1 responded, 2 didn't, or the third one halfway responded at a particular dose. They're not going to give you breakthrough designation. It's not going to happen. And so I think in that sense, if you want to plan breakthrough designation as part of your pathway to approval, you need to rethink your clinical development plan.
And how important is mechanism of action in terms of the process versus clinical data? Because it appears that cell therapy still, we can't elucidate precisely what the mechanism of action is versus in biologics, small molecules, or monoclonal antibodies in particular. It's much better defined.
Well, it's probably a little bit more important because in cell therapies, there's no half-life. It's not something like once you give it, it's going to go away in a few hours. It could be, in some cases, the cells could be there for a lifetime. So FDA is very cognizant, and I think even the advisory committee is also very aware of, they want to know how are these things working. Now, mechanism of action, I think you have to be careful what you're describing as your mechanism of action, because in this sense, manufacturing-wise, FDA has put the potency assay totally based on mechanism of action. And I think that has made the hurdle for developing a potency assay much higher than it is for monoclonal antibodies and recombinant proteins. They don't necessarily say that the potency assay has to be around mechanism of action, where the Office of Cells, Tissues, and Gene Therapy has made that distinction. And it has made it very difficult, I think, for cell therapy companies to come up with a potency assay that the FDA will accept.
So I think I alluded to this before, if I can just jump in here. I think the mechanism of action is very critical when it comes to making process changes. If you don't understand how your cells work and don't have a good potency assay, you run the risk of having the wrong product in the next trial. So I'll say that. I'll also say that for biologics, understanding the mechanism of action is not that simple. Just because an antibody binds to a receptor doesn't mean you understand how it works. So for some of these classical antibodies, it took a long time to get the MOA figured out. So maybe we're not that different in that sense. The difference here is that these cells in the MSC world, the allogeneic world, they're pleiotropic, so they have multiple mechanisms of action. And understanding which one is important for which indication is going to be critical. I think it's very complex. I think, go back to what I said before, the more you understand your cells, the better off you're going to be in terms of clinical success.
David? Mechanism of action? Lonza, how much time do you spend with clients, trying to work out those details in order to deliver a potency assay when you introduce to a new client?
The focus is really not mechanism of action for us. We are looking at it more from, can you separate success from not success? We're looking purely as a manufacturing side. Our challenge is quite often these are so poorly characterized that we don't really know how to get to a potency assay, how to get to a purity assay. And so our focus is really taking and looking at the cells themselves, figuring out what are the markers that are appropriate for it, looking at the indication, and try to pull in the appropriate markers that you can utilize to determine success. In the end, what we want to do is, starting with different master cell banks, get to a point where we're producing the same cell no matter what.
Terrific. Let me see if we have any questions from our audience on it for any of the panel members. So one more question I'd like to ask is just a little bit about how we started the day. We talked about cell therapy being like 10 or 15 years away, and I sat quietly through that because what I see is, well, so for Celgene, you're talking about potentially having a cell therapy in 2019. And as somebody who follows the industry, we know that Mesoblast just started a pivotal trial. We know that there are multiple phase 2 trials about to report out. So I'd like to understand, how you view the timeline on cell therapy, particularly from the Celgene and Lonza point of view.
We view it as, in the next 3 to 4 years, it's going to be an inflection point in the industry. I think there's some other ones that were presented at your presentation in California. I think New Link's coming up on a milestone coming up. There's a couple of other ones that are moving forward into late phase 3 that are really going to drive this.
And from the Celgene point of view?
I agree with that. I think we have a couple of key points this year with clinical data coming out in phase 2 trials, and we'll see if one or more of those are successful. Yeah, I think we're 3 to 5 years away from having something launched.
And from an investor point of view, 3 to 5 years away from commercialization, but maybe 1 year away from proof of concept with some of these results. And I think proof of concept is when you tend to see the valuation inflection where investors begin to believe it's real. Thank you very much. I really appreciate all the expertise on the panel today. Thanks.